Thursday.
Next Thursday.
Okay. I will have office hours on Friday, tomorrow, between 2 and 4. And I think Grace is going to have office hours this afternoon to make up for missing Monday morning. Okay. Her regular office hours will be Monday morning. This week she'll be today between 2 to 4 in room 1204. Okay? So you have those opportunities to talk to somebody if you're confused about anything. Remember, I will not be here next week, Monday through Wednesday, so you will not see me before the exam. And hopefully I won't get snowed in wherever I'm going, so I'll be back for the exam. You will have an exam on Thursday, whether I'm here or not. But anyway, if you want to talk to one of us, those are your opportunities. And then Chanmi will have her usual TA office hours Tuesday morning. Okay, so use us. All right, so I just wanted to kind of review a little bit about voltage clamp and the other thing that we actually haven't named yet. This is where most of the questions are, and this is where a lot of confusion arises. And I guarantee you will be asked about this on the exam, so I just want to kind of go over this a little bit. Okay, so we talked a little bit about voltage clamp, but when we were doing the non-voltage clamp thing, we didn't really give it a name. We were just measuring the voltage, right? And it has a name, and so you can call the thing we can call voltage clamp. The other thing we can call current clamp. And so the idea behind this clamp is that tells you what you're controlling in the system, right? So when we were originally just measuring voltage in the very first part of the lectures, we weren't really manipulating it. Okay, but then I showed you this slide, which is where we started injecting current. So this is actually what we would call a current clamp mode or paradigm. Okay, so clamp is the thing that we're controlling. Can you read this in the back? Did I write big enough? Okay, I gotta get some of those, sidewalk chalk. That'll make it a lot bigger. Okay, so this is what we call a mode. We have current clamp and voltage clamp. Okay, in the clamp part is what you're controlling, and then the other thing is what you're measuring. So in this case, and usually we put what we're controlling sort of in a top graph. So here we're controlling current. So if you're not injecting any current, this just sits at 0. And then we did a bunch of manipulations of injecting negative current, and we noticed that the voltage hyperpolarizes, right? So negative current hyperpolarizes, positive current depolarizes. So that was kind of obvious to everybody, I think. All right, so let's just draw this. I wanted you to be able to compare and contrast this. Okay, so let's see, our next example. So this was, again, this is current clamp. So now we're going to give it a step, and we can measure what happens to the voltage. Okay, control current, measure voltage. So something that looks like this. This is positive, this is negative, and the voltage just basically follows what the current does. Okay, it's very intuitive. Now, when we talked about voltage clamp, right? So now we've flipped it. So now we're controlling the voltage, and in order to measure the current. Now, the confusing part of this is that in order to control the voltage, we're also injecting the current, and that's actually ignored in this graph. So don't get confused by that. So in this case, we're controlling the membrane potential. Okay. So now we're going to voltage. So this is current, and this is voltage, and now this is voltage, and we're going to measure current. Okay. And so notice that the voltage usually has something to do with our resting potential. Usually we start around rest, which is about -65. Okay, something like this. And then you give a step in order to measure the current that occurred because you have changed the voltage. Remember, they're voltage dependent. Okay, and that's what we're looking at here. So there's a bunch of currents that are going on here. We're kind of measuring them. The one that we're ignoring, because I'm going to say it's very, very small relative to the other currents, is the current that we're actually using in the voltage clamp to change the voltage. Okay, that's being ignored here. And what we have is we have one current that is the capacitor charging up. Okay. We have another current, and these are the two that we really want to kind of focus on. So we have this negative current that we call an inward current, and this positive current that we're going to call the outward current. Okay, so let's draw that. Here's our capacitive, inward, and outward. Okay, so this is 0. So out, in. And this is negative, positive. Okay, and this is where things get a little sticky. Remember that we told you that the inward current are positive ions flowing in. So let's see, give myself enough room. So this is, and I know I've drawn this lots of other times. Sorry, it's a little small. So this is sodium flowing in, right? Okay, so why is it negative? And that's a sticking point. I'm going to tell you that it's negative. This inward current is listed as negative just by pure convention. Okay. There is a reason, but I'm not going to really tell you. If you want to know, you can come ask me later. But I'm not going to tell you because it just sort of adds confusing. It's basically a measurement convention. Okay. So this outward current is now potassium ions flowing out of the cell. Okay, so here you have sodium ions flowing into the cell, and here you have potassium ions flowing out. So you just sort of had to pick, right? You had to kind of pick by convention which way is going to be inward, and which way is going to be outward, which one is going to be negative, and which one is going to be positive. We could have done it the other way around. And I think what's confusing to people is that this negative current is actually depolarizing the neuron. Okay, so when sodium ions flow into the cell, it's basically adding positive charge to the cell, like you're injecting positive charge, and so this is depolarizing. And it's just a convention that inward is negative and outward is positive. Does that kind of clarify, or does that make everything worse? Yeah?
So for the inward current, you only see that once you reach the threshold and they go to the action potential?
No, so in the voltage clamp, there is no action potential. Right? Because we're clamping at a particular voltage. Now, the idea is that we're looking at the currents that do underlie the action potential, but we're not allowing an action potential to occur. And that's basically because we're in voltage clamp. We're basically giving an opposite injected current to be able to keep the voltage at the same level. Yeah.
So the negative inward current is basically depolarizing the neuron in voltage clamp, but
it's not depolarizing the neuron in voltage clamp. We're measuring the current.
Yes.
If it was not in voltage clamp, these ions flowing in would depolarize the cell.
Okay.
But it's not actually doing that under these conditions.
How come in current clamp the negative current hyperpolarizes?
The current in current clamp, the negative current hyperpolarizes. So think of it as injecting negative ions.
Okay.
Right.
Thank you.
Okay. Did that help or hurt? How many for help? How many for hurt? Okay, at least one. Well, on balance, then we did better. So if you're still confused about this, come by tomorrow and we'll work through it. Okay. And I just wanted to point out one more thing. So in the very early lectures on electrochemical potential, we showed this kind of a plot. So this was just this simple idea that you have these two compartments and which way are your ions flowing, and we applied a current, we applied a voltage. So in a sense, this is a voltage clamp experiment, right? You applied a voltage, you're controlling the voltage, and you're measuring the flow of ions. So in this case, the ions are flowing in one direction. In this case, the ions are flowing in the other direction. Okay, so it's the same phenomena, essentially. Yes?
In the previous slide, when I was saying something about action potentials not being linear, what does it mean by linear?
Maybe this is slide before this.
Slide before this.
Or is
Oh, here. Yeah, so the idea here is that you can directly predict the membrane potential from the injected current, but once you reach a certain threshold, those voltage-gated channels start to take over. So it's not a direct, right, this spike does not have any direct correspondence to a spiky thing in the current, if that makes sense.
Oh, so it doesn't, B equals IR does not apply.
It does not apply because you've got that positive phenomenon, feedback phenomenon of the voltage-gated sodium channels.
Okay.
So something has gone kind of crazy here. It's not reflected in the original stimulus. Yeah.
Thank you.
All right, let's move on. Okay, so last time we talked a lot about synaptic transmission, right? So we talked about vesicles and action potentials and calcium influx, releasing neurotransmitter, right? Okay, but I wanted to tell you a little bit more about it because it sounded like you covered this a little bit in mammalian physiology, so I thought I'd just give you a little bit more about this and talk about a little bit more about the synaptic transmission. So when we talk about synaptic contacts, oftentimes we're referring to an active zone. So the active zone is really the site at which you have all of those synaptic vesicles, and you have the synaptic cleft, and you have neurotransmitter receptors on the postsynaptic side, and a whole lot of machinery. Okay, this is a very complex biochemical machine, and it's very localized in the brain. And so typically when we're talking about a synapse, we're talking about an active zone. Okay, but morphologically, this can take a number of different forms, and so I wanted to just kind of mention. Maybe I can do a little more drawing. Let's see. Is this going to work? Oh, that's it. All right. So what do these look like? So sort of the most common form of a synaptic contact basically looks like this. Okay, and this is what we would call a bouton, sort of button-like. And so this is where you have synaptic vesicles, and you have neurotransmitter receptors, and lots of other proteins that we'll talk about. And so this is what we mean by an active zone. Okay, and so typically in lots of places in the brain, hippocampus and cortex, this is the type of synaptic contact that we're talking about. But a lot of the lectures that we've given, we've talked about these very specialized synapses. So in particular, we talked about the giant synapse of the squid, neuromuscular junction, and one more is there are these giant type of synapses, particularly say in the auditory brainstem, where you basically have a really big terminal, sometimes onto an adendritic postsynaptic cell. So this is pre, this is the postsynaptic side. And in fact, you have a whole bunch of active zones. Okay, so oftentimes what we're looking at is sort of the summed potential that occurs because of these active zones. And in some of these giant synapses, you have hundreds of these active zones. But they're all kind of acting independently. And oftentimes they have kind of a common pool of vesicles. But at each of these sites, they're acting kind of independently. And by independently, I mean each of those active zones generally has what we call a release probability. So when you have a single active zone, release probability is at a single contact. When you have a giant synapse like this, we usually think of the release probability as being a property of the active zone itself. And they're all kind of acting independently, right? Okay. And generally, at each active zone, when you have an action potential, you have a probability release, and typically you only release one vesicle, even though you've got lots sort of sitting there ready to go. And so this is sort of the one vesicle per active zone dogma. Okay? People for a long time thought that was it. But it turns out, like everything else, there are exceptions. So there's an exception where you have multivesicular release. And these usually happen at very specialized synapses, sometimes in sensory systems, sometimes where you have basically just sort of a lot of continuous release. And so one example is in the hair cells, in hearing, you have these what are sort of physically described as ribbons. So these are ribbon synapses. And you kind of have a little conveyor belt of vesicles. And what these allow lots of release. 2, 3, it's hard to tell exactly how many, but basically multiple vesicles. And that's because you have to have a lot of maintained release. And so there's a bunch of very specialized proteins that are involved in that. One in the auditory hair cells is involved in deafness. And so these are kind of, this is another important mode. But those are kind of the different things. So release probability is kind of a key property of a synapse. So oftentimes we think of a synaptic weight. So this is sort of a synaptic weight. And the release probability, as probability, varies between 0 and 1. And a typical sort of low-end release probability is about 0.01 or 0.1. So every 10 action potentials or every 100 action potentials, it'll actually release a vesicle, which is pretty low. And that's pretty typical of these sort of Bhutan-like synapses. And the basic idea is that in a cortical neuron, you have thousands and thousands of synapses. Remember those first couple of lectures where we talked about, a cortical neuron getting 10,000 synapses? Well, they're all these low probability synapses. And so the point is that you're getting a lot of integration. You need lots of common input in order to activate the cell. On the other hand, when you have something like this, also you typically tend to have a fairly high release probability. But you can imagine if you've got hundreds and hundreds of active zones, you're going to get some release. Even if the release probability is kind of low, you're going to get some release. So these are what we would consider high fidelity synapses, right? You're going to get a response almost no matter what. If there's a presynaptic action potential, very often you get a postsynaptic action potential. It's kind of a one-to-one relay system. So this really has, very strong functional consequences for what's happening at the synapse. And this is where we sort of get this idea of how you get information transfer across synapses. Yes?
So is release probability the probability that one vesicle will be released?
Yes.
Okay. So in the Bhutans...
Sorry, let me repeat that because I know you can't hear in the back. So the question is release probability the probability that you will get one vesicle released per active zone? And the answer is yes.
Okay. So in the one with the one active zone over there, is that a zooming in of the one to the right of it, or is that
Yeah, sorry, these are not quite to scale because this is big.
So that's a zooming in of that.
This is the zoom in. Yeah, so I don't remember what the... Yeah, in the back.
So is release probability, I'm sorry, is it basically the probability just in general or the probability immediately after receiving a signal?
Oh, immediately after receiving a signal. Given that there's an action potential, what's the probability that you're going to get a vesicle released? Yes, so it's, did I write that down? Release probability is the probability, yes, so it's the probability that you will get a vesicle release given an action potential. So it's per active zone, per action potential. Yes.
I'm not sure if I heard you correctly, but is the second picture, is it a zoomed in or is it not?
This is, I don't know. This is big. So say this is, take this.
Oh, that's what that is.
Yeah.
Oh.
Yeah, so let's see. So like this is an active zone, and then this is an active zone. So we have teeny tiny vesicles here. So this is a much more zoomed in picture.
Oh, okay.
The flaw of giving a shot. Yes.
Are there any situations where it is just the Bhutan, or is it always like the big dendrite with a lot of active zones?
If you have a big dendrite, you're going to have lots of inputs.
So do you mean the left picture is part of the right picture, or they're separate things?
They're separate things. These are
In
two
different-
separate examples. Let me think. Let's give this a scale. This may not be completely right. So let's call this 30 microns, and this, 1 micron, couple microns. Yeah. Yes. Yeah. Every synaptic axon probably contacts thousands of postsynaptic cells. Generally, we're going to say this. Let's call this the general paradigm. Okay, so generally, you've got an axon, like in the cortex, that gives lots and lots of inputs. Sends lots and lots of outputs to lots and lots of targets. Every one of its targets has lots and lots of branches where it's getting lots and lots of input. So we're talking on the order of thousands. Let's see, let's call these specialized. Okay. Oops. Okay, so in these specialized systems, all bets are off. So in this particular system, generally you have 3 inputs onto a postsynaptic cell body. So here you've got, like 10,000 to 1, and here you've got maybe 3 to 1. Okay, so those ratios of convergence really, really depend on the particular system that we're looking at. Yes?
So the specialized one, the big one that looks like a Y, that has more integration of information?
So the question is, does the specialized one have more integration of information? Depends on what you consider integration. I would call integration the summation or averaging or collation of lots of inputs. I would think of as integration. This is more of a relay synapse. This is when you don't want to miss that action potential in the presynaptic cell, right? So you have an action potential presynaptic, you get an action potential postsynaptic. And that is really important in certain systems. So in the auditory system, the timing of every action potential is incredibly important. In a few other systems, single action potentials are incredibly important. In cortex, you're trying to get at sort of a bigger view. Right? So you're trying to see what all of my inputs are saying in common, maybe, and get an average. So any one input doesn't really correlate with the output of the cell. We're kind of getting a little off topic, and I find this super interesting, and I'm really glad you're asking questions. That's great. We're kind of going a little farther down these sort of information processing pathways, which is great because I like that. But I have a few other things to tell you about synapses. Okay, so we talked a little bit about calcium, right? We said in our experiment where we were talking about the neuromuscular junction and during quantum analysis, we lowered the calcium concentration, and that reduced synaptic transmission, right? So people said, how is synaptic transmission dependent on calcium? We know it's dependent on calcium. How is it dependent on calcium? Okay, and so they did experiments where you varied the calcium, right? So typically, we're doing our little, we're going to modify the ions in the extracellular solution. And what we end up with, so if we plot calcium concentration versus our, I'm going to call it an EPP, because these experiments were done in the neuromuscular junction, just like we talked about before with quantum analysis. And what they found was that the relationship between the size of the EPP and the calcium concentration was nonlinear. Right? It's nonlinear. It's not a straight line. And it turns out that if you replot this in sort of a log-log plot, you get a relationship with a certain slope that happens to be 4. Very specific. And the implication of this is that basically you need 4 calciums in order to get transmission. And so this leads to this idea of calcium cooperativity. Okay? So you need 4 calciums in order to get proper transmission. Okay? And it's cooperative in the sense that 1 plus 1 is more than 2. Okay? And that's where this sort of nonlinearity comes from. All right. So, and the idea is that, you know, this has to be essentially some power of 4. All right. So what's the calcium sensor? We talked a little bit about, yes, in the back?
I'm kind of confused, like, when you say that 4 need to bind, so I'm confused why there's a graph at all. Like, that would be like nothing, 0, 4, and there's a sudden lump.
So he's saying, I don't get it. How do you get, we need to bind 4 from a slope of 4? Let's see. I knew somebody was going to ask this. And it depends on the kinetics of binding. So I kind of don't want to go into the formula. If you're really interested, I can give you the paper and the formula after.
It's like why 1 on that x-axis isn't just 0.
Sorry, what?
Why like 1 on the x-axis isn't just 0? I'm confused why it's not like that. Because you say 40 is some equation, you might do 1.
I'm sorry, I didn't hear the last part of the question.
Like, you say 4 need to bind, so that to me seems like 3 shouldn't do anything, then 2 shouldn't do anything, then 1 shouldn't do anything.
Oh, yeah. So it's not that 1 doesn't do anything or 2 doesn't do anything. It's that 1 will do something, and you add a second one, it does more than twice as much. You add a third one, and then it does even more. You add a fourth one, and you get sort of your maximal amount. Yeah. Let's move on from that. I don't want to kind of get bogged down in the formula for it. But the basic idea is this kind of cool idea that you can do this log transform and get a slope that tells you something about how the kinetics of this works. Okay, and might make a little more sense. Go on. Okay, so we talked a little bit about all the proteins, the snare complex, and I want to talk about one particular protein that's one of those components, and that's synaptotagmin. Okay, so that's one of those proteins that we talked about, and so people identified a bunch of proteins. They isolated synaptic vesicles and isolated synapses, identified the proteins, cloned them, did expression studies, and so on. In this particular example, they cloned it and then created a knockout mouse. So they had this candidate protein, and when you knock it out of the mouse, it turns out that you get rid of fast synchronous release. Okay, so here's a control. Here's a synaptic response in control conditions. And if you knock out this synaptotagmin and then you stimulate, so assume there's stimulation here, all you get is this sort of delayed asynchronous release. So in order to get that really rapid response of neurotransmitter release, you need to have synaptotagmin. Okay, so it turns out that synaptotagmin, when they cloned it and looked at the structure, that it has these 2 sites for calcium binding. These are called C2 domains. Remember why? So this is similar to another, sorry, I'm forgetting, another protein. Protein kinase C. That's what it is. Anyway, so it has this similarity to another protein whose function was known, where you have calcium binding. And so it turns out that you have 2 of these domains. They're slightly different. One binds 3 calcium, and the other binds 2 calcium. So that's pretty close to needing those 4 calcium. It turns out that more sophisticated models showed that other parts of the curve really depend on 5 calcium. So everything kind of works out. 4 is kind of your lower bound for this system. There's a question.
You said the structure was synaptic, but then the size was syntactic.
No. Oh. Terrible. Thank you.
I'm just.
Yeah, there we go. See? Just too many proteins. Okay, so synaptotagmin 1. And actually there's a whole family of these synaptotagmins. So these are the calcium binding proteins. Okay. And it turns out that these sites bind calcium, and they also allow binding to phospholipids. So when you bind calcium, you promote the binding of phospholipids. And that's partly why you have the fusion activity, right? So synaptotagmin, when it binds calcium, also binds the phospholipids, and that promotes fusion of the membranes. All right. Screw this up on any other slides. All right. So here's another close-up view. This is kind of similar to what we showed before. Here's the other snare proteins. Here's synaptobrevin and SNAP25. And here's synaptotagmin. Okay, so when it binds calcium, it also binds to the membrane. Okay, and there are some other proteins here. The rim proteins that, you know, I'm not really interested in you being able to memorize all of these proteins. I want you to get the concept that there are proteins here that will bind to calcium channels. And so these proteins are not only docking the vesicles, they're also kind of collecting the calcium channels to be very close by. And so what you end up with is what some people call a microdomain. And so you have closely associated calcium channels, and we saw that in the neuromuscular junction, right? You remember all those little dots along the membrane, those are the calcium channels. And then you have the synaptic vesicles that are lined up right along the dots. So everything's organized to keep those calcium channels close. And then you have a local influx of calcium. So it's set up so that the calcium is very close to this calcium sensor. And then a few of these other proteins are also calcium sensitive, so things can get dicey with calcium really quick when you're trying to investigate this kind of thing. But the basic idea, this is really the critical interaction here. Okay? All right, so when you have a local influx, it can rise to tens of micromolars, concentrations. The baseline concentration for calcium typically is in the nanomolar range. Okay. So you're having this really rapid local influx of calcium that's causing that really tight synchronous vesicle fusion event. Okay. And the localization and modification of these calcium channels probably has a lot to do with modulation of function and release probability in the system. Yeah.
So when you say like tight synchronous vesicle release, are you just saying that all the neurotransmitters bind at the same time? Are you, and without that calcium, they aren't able to do that? Because I'm kind of confused by what you're saying with that.
So your question is, what do I mean by synchronous?
Sort of, yeah.
Yeah. Okay, so we usually think of synchronous release. I guess what's missing from this is the stimulus. So here, assume that we have electrically excited the presynaptic axon. And here we're measuring the postsynaptic event.
Yeah.
Okay, and the fact that we have this very large, very short postsynaptic event means that you have immediate release of those neurotransmitter vesicles. Or maybe multiple sites that are adding up.
So would it coordinate between sites? Is that what synaptotagmin does?
So synaptotagmin ensures that the neurotransmitter release happens immediately after the actual event.
Oh, okay. But then how does knocking it out prevent as efficient release? Because the axon potential still comes down, you still get that depolarization, calcium still comes in, right? Wouldn't it still fuse?
Without synaptotagmin, it does not fuse, let's say, efficiently. There's still some fusion. So in this example, there are these delayed events, and you also have spontaneous events, and people are still kind of working out exactly what's causing what we consider these asynchronous events and spontaneous events. And actually, people don't really know exactly what's causing that. Sometimes you can have ambient levels of calcium. So say you have calcium influx, and then you have to sequester the calcium again. And sometimes you have a certain amount of calcium around. You also have to consider that these vesicles are sitting in the membrane, and they're like on a hair trigger, right? And so the influx of calcium sets off the trigger, but you can imagine that you may have other things that set off the trigger. So spontaneous conformational changes, for example. Or you may have spontaneous calcium openings. And there's multiple proteins involved in this. We're sort of trying to divvy it down to just a few. But there are multiple synaptotagmins and multiple other proteins that are all involved that may promote fusion. So.
Oh, okay, so we just know that like it helps with fusion. We don't necessarily understand the mechanism for that.
Yeah, the mechanism, so, yeah, mechanism can, we could continue to go down this mechanism. So it depends on what you're counting as a mechanism. So
I was just saying, like, so within one vesicle, right? You're talking about synaptotagmin, it helps that one vesicle release all of its neurotransmitter contents efficiently.
Yeah.
Whereas without it, you have sort of this haphazard fusion where some neurotransmitter leaks out here and there in a way.
More or less, yeah.
So it's delayed. Okay, so then do we understand how that happens?
No.
Okay. It just happens. Okay.
No. I mean, it's still under really active investigation. We don't really know why release probability is different at different active zones. It may be different with different vesicles. There are numerous steps in preparing a vesicle for release, and you could modify things at any point along those steps. Yeah, but it's complicated. But in that, yeah.
So, is it something that we just kind of that like for the purposes of class we simplified that it seems like synaptotagmin, from what we've seen, is the only like calcium dependent thing in the whole adoption
It
process?
is not remotely the only calcium dependent thing in this whole process. Many of these steps are calcium dependent. Recruitment of vesicles, don't have time to talk about this today, but there are multiple pools of vesicles. So remember that vesicle cycling? That can all be calcium dependent to some extent. Yeah, and the synaptotagmin is really important sort of at this final exocytosis step. But let's see, hang on. So this little diagram, shows that this protein has a calcium binding site. This protein might have a calcium binding site. It's all over the place. And this is why it's so difficult to kind of peel apart what's doing what. Because you have multiple proteins that have multiple calcium dependencies at multiple steps. It's a difficult problem. Yeah. And we could knock out one of the synaptotagmins only to find that there's another one that could take its place and do just as well. So it's a tricky problem. Very tricky.
So when the synaptotagmin binds calcium, it then promotes phospholipid binding. When we say phospholipid binding, do we mean phospholipid binding to calcium or phospholipid binding to when we're talking about like fusion of the vesicle to the membrane?
So the idea is that the synaptotagmin, that these sites also bind phospholipids.
The synaptotagmin also binds
The
phospholipids?
synaptotagmin, well, there's this membrane crossing region here, and that's what inserts it into the vesicle.
Mm-hmm.
But these C2 domains also can bind phospholipids. And so the idea is that if they bind calcium, that's going to promote that these are going to want to bind to maybe the plasma membrane.
So the synaptotagmin is already in the vesicle, and when it finds the calcium, then the other end of the synaptotagmin binds into the membrane, which is then helping you get those.
That's the model.
Oh, okay.
Yeah. Remember, a lot of these things you can't see, and you have to kind of infer. And so you kind of build up this model of how these proteins interact. So I'm not showing this. So you can do an experiment where you can take synaptotagmin and run a bunch of phospholipids over a column, and when you add calcium, you get more phospholipid binding. So that's kind of the basis of these bind phospholipids. Now, how it actually binds, does it actually bind the plasma membrane in this system? It's something that's hard to see. It's hard to prove, but that's the model.
Thank you.
Yeah.
Makes sense.
All right. All right, I think that's it for that. Okay, there's a lot more to know, but I think we're going to move on. All right. Okay, so we've talked a lot about the presynaptic mechanisms. We could go on for another couple of days about presynaptic mechanisms, but let's move on to the postsynaptic side. Okay, and we're going to talk about ligand-gated neurotransmitter receptors, excitation and inhibition, and a little bit more about synaptic integration, sort of from the postsynaptic side. And then we're going to start talking about neurotransmitter systems. We're not going to get anywhere close to through that, so this is basically chapter 6. Kind of boring for me to stand here and tell you about this neurotransmitter and that neurotransmitter. You can read a lot of this, and so I'm basically going to assign you to go through chapter 6. I'm going to post the notes from this lecture. Whatever we don't get through, just continue on your own through chapter 6, and you can follow my notes as a kind of guide for what you should focus on. Okay. All right. So the basic gestalt here is that we have neurotransmitter release that leads to postsynaptic receptor binding. Okay, that typically leads to ion channels that open. Okay, these may be one and the same thing. We'll show that. Then you have a conductance change that causes current flow that results in postsynaptic potential changes, which can lead to either excitation or inhibition. If you're excited enough, you can get an action potential. Okay, pretty straightforward. Okay, and so the sort of star of the show, in terms of postsynaptic responses, are the ligand-gated ion channels that are what we call ionotropic. These are ion-passing channels. So when a neurotransmitter binds, there's a little gate that opens, and you have neurotransmitter that goes through. And we're going to first talk about the acetylcholine receptor, abbreviated ACHR, sort of the canonical excitatory neurotransmitter. Okay. But ionotropic, that's the key word. All right, so similar to our experiments with voltage-gated currents, we're going to start to look at these ligand-gated currents. Okay. And so first we can look at single channels. So just like the voltage-gated channels, we can express ligand-gated channels in our expression system, make patch recordings from small patches of membrane, and look at single channel openings. And that's what we're looking at here. So this is, again, this is always done under voltage clamp, so we're looking at currents. Although these plots, for some reason, so these plots are saying number of open channels, but you could read this as a current. Okay, so 0, channel opens, we get some current, and then it closes. Okay, just like with the voltage-gated currents, if we add up a whole bunch of them, we start to get something that looks like this. And macroscopically, if we look at a synaptic current, it looks like this. So when you have a when you have a neurotransmitter release, you get something that looks like this. So this, you could read this as a current, and it has a particular shape that we would call an alpha function, a geeky name for this shape. But this is sort of characteristic of these synaptic potentials or currents. Characteristic of the synaptic current, and then the synaptic membrane potential basically kind of follows the current. Okay. Couple of abbreviations that I will use all the time without thinking about it. Excitatory postsynaptic current, EPSC. So that's what you measure on their voltage clamp. And then excitatory postsynaptic potential is the voltage response, EPSP. Okay. And this shape is really reflective of the, more or less of the kinetics of how the ligand binds and unbinds the channel.
So that's the alpha function, number 3. Okay.
Okay, and just like our voltage-gated channels, ligand-gated channels have a reversal potential that's reflective of its ion permeability. Okay, so we call this a reversal potential. Okay, and when neurotransmitter arrives, so just like we learned with the sodium channel permeability, that if you open a channel, that the voltage tends to go toward that equilibrium potential, that channel. Okay, so when you have neurotransmission, essentially the postsynaptic voltage is going to move toward the reversal potential for our ligand-gated channel. Okay, so obviously it depends on what ions the channel is permeable to, right? Okay, so for excitatory synapses, it tends to be permeable to sodium or calcium, or it's what we call cation non-selective, sodium or potassium. Okay, and typically the reverse potential, just like when you learned sodium has a reversal potential of, anybody? Plus 40, plus 50, somewhere up there, so really high. Okay, so it's this reverse potential is always high or greater than or equal to 0. Okay, and that's going to push your voltage in a depolarized direction, right? Inhibitory synapses, on the other hand, tend to have either solely potassium or solely chloride, and those have very negative reversal potentials around your resting potential, right, or less. And so those tend to cause the membrane to move in the negative direction, which is hyperpolarizing. Okay, and that's the sense in which one is excitatory, pushes the voltage up, the other is inhibitory, pushes the voltage down, right? And all of this is relative to threshold. Okay, so let's look at some examples of what we mean by reversal potential. Okay, so let's say we're measuring, this is again like frog neuromuscular junction. Here's your muscle. We're doing a voltage clamp experiment, so we have 2 electrodes that are clamping the voltage. We're going to measure the current. Okay, so what we're measuring is the EPSC current. And we're going to stimulate the presynaptic axon. So a lot of times we sort of don't indicate that the stimulation is happening, but that's shown here. So stimulate the presynaptic axon. After a little bit of delay, we get a nice negative current. Okay, what did we say about negative currents? Are they inward or outward?
Inward.
Inward. So what happens to the membrane if you have an inward current? It depolarizes. So in that sense, it's excitatory. Okay, so an inward current essentially is excitatory when we're sitting at sort of our negative voltages. So if this is at a negative 110, you get a big inward current. At negative 60, you get a slightly smaller inward current. At 0, there's no current. What does that tell you? At 0, there is no current. Are ions flowing? No. Well, no net flow, let's say. There's no net flow. So this is what? What would we call that point?
That's the equilibrium.
You want to say it?
Equilibrium.
Equilibrium, yes. So basically
Potential.
an equilibrium potential for this ion, what we call a reversal potential. And then, lo and behold, you depolarize more, hold the voltage at a more depolarized voltage, you get an outward current. So in this sense, it has reversed. You've reversed from inward to outward across the what would be the equilibrium potential. Yes?
So
Sorry, louder.
The reversal potential is
For that ion channel. Yeah, let's look at that. Okay, so let's plot that data. So this is the same experiment, and now we're plotting the EPSC amplitude. So we found that at low voltages, we have this negative inward current that then gets smaller as we depolarize. When we get to 0, there's no flow, so there's no net current. When we go to more positive potentials, now we've flipped it, and now we have an outward positive current. Okay, so this is what we would consider our IV curve. Okay, and notice that where it crosses is the reversal potential for that ion channel. Okay, now I want to remind you the reversal potential, so the Nernst potential for potassium is about here. For chloride, maybe about here, depending on exact concentrations, but roughly. Sodium is positive, it's way up here. So 0 is kind of neither of these things. So how can we tell what's causing this? Well, let's make a bunch of plots that if the reversal potential for this ion channel was equal to EK, you'd expect a linear plot like this. If it were equal to ENA, we'd expect a plot like this. So this is the reversal for ENA. If it were chloride, it'd be something like this, and it's none of those, right? So how are we going to tell what ions are being carried in this channel? So let's do an experiment, okay? So here's our original IV plot. It goes through 0. 0 millivolts gives you 0 current. And now let's do our ion concentration manipulation, like we like to do. Okay, so we're going to modify the external ions and see what happens to the synaptic IV curve. Okay, if we lower sodium, external sodium, that shifts our reversal potential, our Nernst potential for sodium in the leftward direction. So it gets lower, right? Okay, and what happens to our synaptic IV? It shifts to the left. Okay, so anytime you manipulate an ion concentration and you see a change in your measured response, you can infer that that ion has something to do with it, right? At a simplest level. Okay? So let's do another experiment where we increase external potassium. And when you increase external potassium, the Nernst potential for potassium shifts to the right. Okay, and that's what we see. So here's our original control synaptic IV. And when we increase external potassium, it shifts to the right. Okay, so from this, we can infer that both sodium and potassium contribute to the ion flow through this channel. Okay, so we're getting a reversal at 0 in our control conditions. Lowering external sodium shifts the curve in one direction because ENA goes from something positive to something less positive, and then the reverse with potassium. If you shift EK, you can shift the reversal potential for the synaptic current. Okay, and let's just say that modifying chloride doesn't do anything. We can kind of eliminate that as a contributing factor. Okay, we can conclude from this that the acetylcholine receptor is a non-selective cation channel. Okay, it passes both sodium and potassium, and it's kind of roughly equal. Okay, and that's why you end up at about 0. Okay, so the permeability of the acetylcholine receptor is roughly sodium and potassium. And you might ask, well, what about calcium? Right? We have a really big calcium gradient. And it turns out that calcium does, in fact, go through this channel. So a lot of these ligand binding channels, excitatory channels, pass some amount of calcium. But they basically kind of prefer the monovalent ions. But some component, about 2% of this is actually passed by calcium. Okay, so let's talk in the last 15 minutes or so here about excitation and inhibition. So we just talked about acetylcholine as an excitatory receptor. And so whether something is excitatory or inhibitory really depends on what effect it has on the membrane. Okay, so we're going to, I told you before that EPSP stands for excitatory postsynaptic potential. Likewise, IPSP stands for inhibitory postsynaptic potential. And, you know, we talked a lot about acetylcholine, but really in the mammalian brain, central nervous system, glutamate is really the major excitatory neurotransmitter. Acetylcholine is very much a peripheral, muscular neurotransmitter, but in the brain, it's all about glutamate. Okay? And the major, probably the major inhibitory neurotransmitter is GABA. You've probably heard about these, glutamate and GABA. Okay, can you see that? Let me turn down the lecture. I don't know why they choose yellow. I never use yellow in my slides. Okay, so just to kind of give you an overview, this is all very intuitive, I think, that when you activate a glutamate synapse, so here we're just measuring the voltage, right? So we're in current clamp. Okay, and we're measuring voltage, time is on the x-axis. Okay, and you activate a presynaptic synapse that happens to be glutamatergic, and you can measure an excitatory potential. Okay, if this excitatory potential is big enough that it reaches what we would call threshold, we get an action potential. Okay, so here your membrane potential is sitting at rest. The reversal potential for this excitatory channel is up here at 0. So the idea is that when you open those channels, that you're pushing toward the 0. You may not get to 0, but you're pushing it in that direction. And if you do it sufficiently, you can get an action potential. And that's the sense in which this is excitatory. Anything that pushes you closer to the threshold is excitatory. Conversely, if you activate a GABAergic synapse, you get an inhibitory current. An inhibitory potential here. So when you activate GABA synapses, we'll talk about this in a minute, GABA has a negative reversal potential, and so you're pushing the membrane in the negative direction. In that sense, you're moving away from threshold, it's inhibitory. Okay, and that's generally what I just said. So what happens if you have different types of inputs coming in at the same time? So say here's a neuron, you have some inhibitory inputs, and you have some excitatory inputs. What's happening in the postsynaptic membrane? Okay, so here's a little plot of what happens when you stimulate these different pathways. I'm just going to, in the next slide, I'm just going to expand this, but this is the experimental setup. Okay, so if we excite a single excitatory input, we can get a small EPSP. Okay, if we excite 2 of them, they add up. And if they add up sufficiently, they can reach threshold and give you an action potential. If you stimulate an inhibitory, you get an inhibitory potential. If you stimulate excitatory and inhibitory, they tend to add. In the sense, once one's negative, one's positive, they kind of cancel each other out. Yes?
But just to be clear, an excitatory stimulus creates a negative current but a positive change.
Yes, and that's not here. So yeah, so that's the confusing part about the voltage clamp. And inward current, by convention, is listed as negative, but it actually has a depolarizing effect on the cell. Yes?
When you talk about single excitatory input, what molecular measures are, I mean, it's probably all unapplicable then, right?
So in this particular example, we're in current clamp. So we're not actively injecting current. We're measuring the voltage effect of a current influx. So you have to kind of imagine that when you have neurotransmitter release, you're opening ion channels. When the ion channels open, current flows in. Current has an effect on the voltage, and what we're measuring here is that voltage effect.
So when you talk about a single input, you're talking about X
imagine, just one of these contacts.
So it's like one. What's the X in that case? It has to be something that's formalized, right?
What do you mean?
So you're releasing a certain amount of neurotransmitter each time, otherwise you wouldn't be able to figure out what's excitatory, the excitatory and inhibitory.
Yeah, so I'm gonna kind of elide the how much neurotransmitter, how many vesicles, because oftentimes when you do these kinds of recordings, you're recording these some inputs. For simplification, we're just showing a single input. But so we're showing a single input, but you could imagine that this would be a small set of inputs or a single axon that has multiple active zones. I don't want to get too technical about this. This is just, yeah, come talk to me after. Yeah. Okay, over here first.
So is it an action potential that stimulates for an IPSP in the first place? I'm just
Yes.
looking, okay.
Yeah. So these are axons, okay, and there's an action potential coming down the axon, and if you kind of blow this up in your head, the action potential invades the terminal. It causes exocytosis, which releases neurotransmitter, which crosses the cleft, which binds the ligand-gated neurotransmitter receptor, which opens its channel, which allows ions to flow, which is an inward current in the case of excitatory, and outward current in the case of inhibitory. That causes a potential change in the postsynaptic cell. If you have an inward current, it depolarizes, and that's what we're seeing here.
Okay, thank you.
Yeah. Yeah, so there's a lot that goes on between the action potential in the axon and our measurement in the postsynaptic cell. Yes.
So what determines whether it's excitatory or inhibitory?
What determines whether it's excitatory or inhibitory? Okay, well, a couple of things. Which neurotransmitter is released? We said that glutamate is a neurotransmitter that has an excitatory effect, and GABA is a neurotransmitter that has an inhibitory effect. But the reason it has an excitatory or inhibitory effect depends on the postsynaptic receptor. And we're not going to quite get to this today. Okay, maybe we'll do one more lecture on neurotransmitters after this. Okay, so the reason that this is excitatory is because glutamate as a neurotransmitter binds a ligand-gated receptor. We're gonna get to this in a little bit. I just want to answer your question right off. Is it binds a particular receptor that has a reversal potential of 0. Okay, and we're going to talk a lot more about this in the next couple of lectures, so don't worry too much about this. And then GABA is a neurotransmitter that binds a different neurotransmitter ligand-gated receptor that passes ions such that it has a reversal potential that's very negative. Okay. And that's why you're pushing the membrane either depolarized or hyperpolarized. It depends on the receptor. Yes?
So when you say, so if there is a specific receptor with a reverse potential, when the receptor opens, the channel tries to reach to its reverse potential?
Yes.
Okay.
Yes. So the question was if you, this is tricky, rephrasing it. So the question was if you have a ligand binding receptor and you open the receptor, the membrane wants to go to the reversal potential for that receptor. Yes?
So what happens if glutamate binds a receptor with the negative reversal potential?
It doesn't.
It just won't.
Yeah, let's hang on. So these are great questions, and they're going to be answered in due time. But for now, I just want to kind of focus on this idea that you can have inputs that cause depolarization and inputs that cause hyperpolarization, and they kind of sum up. Okay, at a very simple level, they kind of sum up. If you have something that's both positive and negative happening at the same time, they kind of cancel each other out. Okay, if they're equal and opposite, then you get a cancellation. Okay, And that's why excitatory, a lot of times we'll talk about a balance between excitatory and inhibitory, because that really determines whether these inputs have a net excitatory effect or a net inhibitory effect. If you have more excitatory and less inhibitory, you can still get activation. If you have lots of inhibitory and not enough excitatory, you won't get activation. This is sort of a very simple lesson. Yes?
Is it like a second current?
Yeah, the last one here?
The
This-
second one from the left.
The
Like
second,
the-
oh, here,
Yeah.
E2 plus E1 plus E2, yeah.
The ligand and gated channel open throughout the entire action potential, or does it close? Does it deactivate?
Yeah.
Second current makes it look like it's kind of fine.
Yeah, so when you measure the voltage, so what would happen if you like blocked the action potential? Let's ask that question. If you block the action potential, say block the sodium channels, you would see the underlying shape of the synaptic potential. So sort of underlying here is that synaptic potential. And it's just being obscured because you have, now it's taken over by that voltage gated effects. There are some kind of sophisticated effects of the voltage on the synaptic potential, but we're kind of not gonna get into that. Yeah, but sort of underlying that that action potential, you could imagine that synaptic current is continuing on. Yes?
So that's the difference between like a graded potential and the action potential, correct? So like the graded potential is what's coming in, and then if like at the summation point, then if it like hits the threshold, then you can have an action potential.
Right.
But you're still going to have like in the dendrites, you're still going to have a change in potential.
Yeah, that's very, that's exactly right. That's very insightful. So these synaptic potentials are an example of what we talked about as graded potentials in the first couple of lectures, right? So graded in the sense that this amplitude could be anywhere from very, very small to very large. And then with the action potential, you have that all or none effect. Okay, and so this is kind of a really sophisticated question here. So you have this dendrite. So say you've got a really big dendrite, and you have these inputs, and you have all of these graded potentials, kind of summing out the dendrites, and then they're passively flowing towards the cell body. When they reach the cell body, you basically have complete summation, including whatever synapses are contacting the cell body. And then at the axon hillock, you have that spike initiation zone. Okay, so if everything gets down to the cell body and the axon hillock, and you've reached the appropriate voltage that we're going to call a threshold, then at the axon hillock, you'll get an action potential. But yeah, so a lot of the summation is kind of going on out in dendrites. Yeah. And then there's some business about whether action potentials backpropagate into the dendrites, but let's not go there. All right, let me just summarize. Okay, this whole concept of synaptic integration, and this is something that is really still being worked out as to whether everything just sums, whether you have nonlinear effects of excitation and inhibition in dendrites, whether action potentials backpropagate and affect this summation. But at the very simplest level, just think of it as basically a summation of those graded potentials that are a result of the synaptic currents. Okay. So one distinction we kind of want to make a little bit, I don't want to overemphasize because every neuron, every system is a little bit different. But we can usually think of the excitatory inputs as being kind of distributed all over the dendritic tree. This is kind of the canonical view. Inhibition can also be on the dendrite, but oftentimes you find inhibition at the cell body. So what do you think the effect of having sort of like a big, strong inhibitory synapse at the cell body is going to have? Yes?
Turn off the action potential.
they can turn off the action potential. So whatever is happening out in the dendrites, if you have a strong enough inhibition at that point of summation, you can basically veto whatever activity is going on. And so that seems to be a very common motif. A lot of inhibitory interneurons seem to contact either the cell body, or there's one that sort of wraps around the axon hillock. So where you put these synapses really can affect how information is being integrated in the neuron. Okay, so currents summate to produce an overall rate of potential change transmitted to the cell body and the axon hillock. Okay, and the end point is either the sum potential will either depolarize the membrane. If it crosses threshold, you get an action potential. If it's hyperpolarized, then you're going to inhibit the cell. Okay, and as we started to allude to, just to finish up, this is the last slide of today, the effect of this neurotransmitter really depends on the receptors. I mean, yes, it depends on the neurotransmitter, but as we'll see in the next lecture, the neurotransmitter has partner proteins, the receptors that are specific for them. Neurotransmitter has family of receptors, and then what those receptors do really determines what the effect of the neurotransmitter is. Okay, the same neurotransmitter can have multiple target receptors, and those receptors could have potentially different effects on the cell. Okay, we're going to talk about when we talk about ionotropic. We're going to talk about secondary indirect effects. The non-ionotropic, metabotropic receptors next time. And so next time we're going to talk about neurotransmitters. Read over